Hello, my name is Amy Elias, and I'm an application scientist for Roche Diagnostics. I'm excited to present to you our new streamlined workflow for a shorter KAPA RiboErase and RNA HyperPrep. Today we will discuss the basics of transcriptomics, followed by the protocol for our standard KAPA RiboErase and RNA HyperPrep. I will then discuss the changes made in the standard protocol to create the shortened KAPA RiboErase and RNA HyperPrep. I am very excited to then showcase our data comparing the new shortened protocol to our standard protocol and discuss the benefits. RNA sequencing is conducted by converting RNA into cDNA, which is then adapter ligated and amplified. The final adapter ligated cDNA library is then sequenced. Using our robust KAPA RNA HyperPrep kit, you can gain insights into gene expression data, alternative splicing information, fusion detection, and single nucleotide variant detection. One common problem in the RNA sequencing field is ensuring that your protein coding transcripts are sequenced at the proper depth to gain these insights. Since 80% of transcripts are ribosomal RNA, or rRNA, depleting these transcripts can both reduce the sequencing cost by not wasting reads on rRNA and allow for more coverage of protein coding sequences. To conduct ribodepletion, we use our KAPA RiboErase kit. KAPA RiboErase is for use in human, mouse, and rat, FFPE, or high-quality RNA. To begin, 25 nanograms to 1,000 nanograms of RNA is hybridized to depletion probes specific for rRNA. The rRNA is then removed with an RNase H digestion. Following a pure bead cleanup, DNase then digest the probes. After the probes are digested, a pure bead cleanup is conducted and the resulting solution contains your ribodepleted RNA. In total, this process is about 2.5 hours. Following ribodepletion, the ribodepleted RNA is then input into the fragmentation and priming module of KAPA RNA HyperPrep. RNA is fragmented according to desired library size, which is dependent on both input type and quality. For example, FFPE and high-quality RNA have different fragmentation times and temperatures to achieve a 200 to 300 base pair fragment. The fragmented and primed RNA then undergoes first-strand cDNA synthesis, whereby the RNA is reverse transcribed into a strand of cDNA. Second-strand synthesis and A-tailing immediately follows, whereby the complementary strand of cDNA is transcribed and marked using uracil. By marking the second strand with uracil, the first and second strand of cDNA can be distinguished and thus creates a stranded library since the uracil marked strand will not be amplified. Additionally, during this step, the poly A is added to the cDNA. This poly A tail is then used in the next step to aid in ligation of adapters compatible with Illumina sequencers. KAPA UDI adapters are recommended for this workflow. Once the cDNA library is adapter ligated, it undergoes 2 bead cleanups. This library is then amplified and undergoes a final 1x bead cleanup. Once this 4-hour process is complete, you have efficiently created adapter ligated cDNA libraries. Although our current KAPA RiboErase and RNA HyperPrep kit has robust performance, we found that there are some steps that can be more streamlined to make this a more efficient protocol without sacrificing data quality. During the KAPA RiboErase protocol, DNase digestion can be shortened by 15 minutes. During the RNA HyperPrep portion, our first modification is made during first-strand synthesis. Primer extension during first-strand synthesis occurs at 20 degrees Celsius rather than the original 25 degrees Celsius. During second-strand synthesis, the protocol has been shortened to 5 minutes at 42 degrees Celsius, followed by E-tailing at 62 degrees Celsius for 10 minutes. This allows for second-strand synthesis and E-tailing to be reduced by 25 minutes. Although the adapter ligation incubation step remains the same, the adapter concentrations for initial inputs between 25 nanograms and 49 nanograms is now 0.15 micromolar rather than 1.5 micromolar. All other adapter concentrations remain the same. after adapter ligation, both hands-on and turnaround time are both reduced via consolidation of bead cleanups into a single 0.7x bead cleanup. All other steps remain the same, and the changes made on this protocol overall lead to about a 1-hour reduction in time. By consolidating a bead cleanup, we also reduce our hands-on time and plastics usage. To test this new shortened workflow, 25 nanograms and 100 nanograms of FFPE RNA were used to test FFPE conditions, while 25 nanograms and 250 nanograms of high-quality RNA were used to test the high-quality input conditions. When we look at our data, we can see that the shortened and standard RNA workflows produce equivalent genomic alignment. On the y-axis, we have the percent of aligned reads to the human genome, and on the x-axis, we have the alignment for the 25 nanogram and 100 nanogram inputs for FFPE RNA on the left, and 25 nanograms and 250 nanogram inputs for high-quality RNA on the right. The shortened RNA workflow is shown in the blue, while the standard workflow is shown in the gray. From this graph, we can see there's no statistical significance in the genomic alignment rates between the standard and shortened workflows for both FFPE and high-quality RNA inputs. We next wanted to ensure that ribodepletion would not be affected by the 15-minute difference in DNase digestion. Here we show that the percent rRNA contamination on the y-axis from 0% to 1% in increments of 0.25%. We then have our 25 nanogram and 100 nanogram inputs of FFPE RNA on the left, on the x-axis, and 25 nanogram and 250 nanogram inputs of high-quality RNA on the right. The shortened workflow is shown in the blue, while the standard workflow is shown in the gray. From this, we see there is low rRNA contamination in both standard and shortened workflows, and no statistically significant differences between the two for any input mass or input types. To ensure the consolidation of bead cleanups and changes in various incubation times and temperatures did not change the number of transcripts detected, we plotted the number of transcripts on the y-axis and the input masses on the x-axis. The FFPE RNA inputs are shown on the left, while the high-quality RNA inputs are shown on the right. The shortened workflow is in the blue, while the standard workflow is in the gray. Here we see that we have over 60,000 unique transcripts in the FFPE inputs and over 70,000 unique transcripts in the high-quality inputs. There is no statistically significant difference between the standard and shortened workflows for either input type or input mass. Less than 5 genes are differentially expressed between the standard and shortened workflows out of the over 30,000 analyzed between the conditions. With the elimination of a bead clamp, we also wanted to ensure that our GC content distribution did not deviate between the standard RNA workflow and the shortened workflow. On the y-axis, we have the fraction reads, and on the x-axis, we have the GC content percentage. Blue lines confer to the shortened workflow, while gray lines confer to the standard. The top plots are for FFPE inputs, while the bottom are shown for high-quality inputs. As shown, there's little deviation between the two workflows. Therefore, the consolidation of bead cleanups did not cause differences in the GC content distribution. As was shown by the data presented and other data not presented here, the shortened RiboErase and RNA HyperPrep workflow yields equivalent results to the standard RiboErase and RNA HyperPrep workflow. There's equivalent genomic alignment rates, equivalent rRNA depletion, equivalent transcript detection, and equivalent GC content distribution. The benefits of using the shortened workflow include reduced turnaround time by over 1 hour, reduced hands-on time via the consolidation of a bead cleanup, and reduced plastics usage. Furthermore, these benefits are achieved without sacrificing quality data. Thank you, and for any questions or inquiries, please contact our support team at the following phone number and email. We look forward to hearing from you.